Can anyone tell? Hello?
Increase leukocyte
So
count above
leukocytosis is
10.
increase in WBC
Yeah, how much?
above the normal range.
Above?
Above
Okay.
11,000 per micro
So in
per
adults,
mm.
it's above 11,000 per mm cube. There can be physiological causes and also pathological causes for increased TLC. So can you name some physiological causes leukocytosis? Like normal conditions in which
Sir,
WBCs are
at
raised
birth
above.
and at birth and pregnancy.
Okay, various like birth, pregnancy, high temperature, severe pain, and muscular exercise. TLC can be raised, and in various pathological conditions like infections, malignancies like leukemia, and also during severe hemorrhage. What and leukopenia is TLC count less than 4,000 cells, and it can be seen in infections like bacterial, viral, or protozoal, and also in megaloblastic anemia or bone marrow depression, as seen in aplastic anemia, which you just read. Now, the various methods to count WBC is first one is visual hemocytometer method, and second is electronic method. Today, we are going to study the visual hemoglobin hemocytometer method. So, hemocytometer, hemo means blood, cyto means cell, and meter means measurement or counter. So, we can use either EDTA blood sample or a capillary blood sample for the count of WBC. And what all do we require? We require a microscope, hemocytometer, WBC diluting fluid, and we also require a Newberg slide, cover slip, and a WBC pipette. So this is a Newberg's chamber, as you must all be aware. This is a WBC pipette and this is a RBC pipette. And how do we differentiate WBC pipette and RBC pipette? Can anyone tell me?
Sir,
So,
So on the
WBC
okay,
basis of
pipette
firstly we see
is
the
marking.
Marking
marking. So,
HMOCA.
the marking is
One by
that in
one by
WBC,
one person at a time.
ma'am, there are three basic features that we can use to distinguish. Number one, it is the bead color, that is red in RBC, white in WBC. The second thing is the
Okay.
marking after the bead, which is 101 in RBC and 11 in WBC. And
Okay.
yeah, I think these were the two only.
Okay. Now, what is this? This is a Newberg slide. And what's the difference between a Newberg slide and a Newberg chamber? A Newberg's, when we cover the Newberg slide with a cover slip, it's known as a Newberg chamber. So it is a thick glass slide, Newberg slide, and in the center of the slide there is an H-shaped groove. Like this is the H-shaped groove, and on the two sides of the central horizontal bar there are scales, and here we have scales for measuring the cells. And let me show you in another pic. Like these are the scales where the counting of WBCs, RBCs, and platelets is done. These are the two vertical gutters, and this is one horizontal gutter. Okay. So when we place the cover slip, the depth of this is zero point 0.1 mm. Okay. So the counting chamber is divided into nine large squares. Okay, these are the nine squares, and the length from here to here is 3 mm, and each large square is having 1 mm length and 1 mm breadth. Okay, so this is 1 mm, 1 mm, 1 mm. Here also 1 mm, and whole this is 3 mm. So WBC counting is done in these four large squares, each having length and breadth of 1 mm. When the Newberg slide with a cover slip over it is called a Newberg's chamber, and we use the four large squares for counting the WBCs, and there are total, and these four squares are further divided into four smaller squares like these. These are the four smaller squares in which the large squares are divided. And this is a WBC pipette. It has a white bead and it has graduation mark up to mark 11. It is the mark 11. 0.5 mark, we have 1 mark, and 0.11 mark. So uh, what's the diluting fluid used in WBC estimation, WBC count? Can anyone tell? Mm,
The tox fluid, which contains Jensen violet and acetic acid. Jensen violet is 1%, which is for giving them color, and 0.3% acetic acid, which is for them to stick to the surface and to kill the microbe. And for the RBC as well,
uh,
because they can.
yeah.
So they're mainly for the RBC because they can also appear on the surface. So to avoid RBC, so we use glacial acetic acid.
So how does it avoid RBCs? Like what does it do?
It
It's just
kills
a
them.
lysine fluid. It lyses the RBCs.
Kills them.
So it lyses the RBCs. So we use Turk's fluid as a diluting fluid, and it consists of glacial acetic acid, gentian violet, and distilled water. Glacial acetic acid is used to lyse RBCs. Gentian violet is used to stain the WBC so that they can be easily visualized. And all these are mixed with distilled water. So this is the principle. Glacial acetic acid lyses the red cells, while gentian violet stains the nuclei of WBCs, and the blood specimen is diluted to 1:20 in a WBC pipette with diluting fluid. The cells are counted in a low power, which is 10x of the microscope, by using a counting chamber, which is a Newbus chamber, and the number of cells in undiluted blood are reported as, and this is how we report, as cubic millimeter per microliter of whole blood. So this is the procedure. We draw blood up to the zero point 0.5 mark. This is the zero point 0.5 mark. We draw blood up to this mark of a WBC pipette, and we carefully wipe the excess blood, which might be at the tips of outside tip of the pipette, so that it doesn't interfere with the results. Then we draw diluting fluid up to the 11 mark. This is the 11 mark. We draw the diluting fluid up to this mark, and then we mix the contents of the pipette by rotating it 2 to 3 times. And after 5 minutes, we discard the few drops, like we discard up till here, because it contains only the diluting fluid. Because first we take blood till here, then we take the diluting fluid. So the blood along with diluting fluid comes along here, and in the initial zero point 0.5 is, or initial up to 1.5, is only diluting fluid. So we discard that initial few drops, which amounts to approximately zero point 0.5 units. Then we allow the cells to settle for two. Then we do the charging of the Newbus chamber. How we do it? We place the cover slip over the Newbus chamber, then along the side of the Newbus chamber at the 45 degrees angle, we drop a drop so that it covers the whole area. This, and we try to avoid excess fluid by preventing the flooding, or we also prevent underloading or overloading. This is known as overloading, and this is known as underloading. So we try to prevent this, and then we wait for 2 to 3 minutes so that the cells can settle over the chamber. Then we use 4x to first of all, we place the Newbus chamber on the microscope, and we, you know, with the 4x, we focus it under the microscope so that it's easier to count the WBCs. When the center, the squares are focused under the 4x, then we charge the Newbus chamber, and then using the 10x of the microscope, we do WBC counting. So how do we do counting? Can anyone tell how WBC counting is done,
So,
and in which squares
so it
it
is
is done?
in an
In
inverse
the
L
bigger
rule.
What?
So we use the
Speak.
inverse L rule.
Okay. And what is
In which
that?
basically, like an inverse L is to be memorized in our mind, where we take a horizontal upward and basically L ka ulta kar diya. So the cells which are on the upper margin and on the left vertical margin, they are counted to be included in the consecutive cell. And if they are
Yes.
like, say the two cells are together, so the one which is on the interface, that will be counted in the second cell and not the first cell, and it's in a
Yeah.
zigzag manner.
So, like, this is the square in which WBC counting is done. This. So let's magnify this. Like, this is the magnified version. So if the cells are present in the bottom line or right line, these are not counted. Bottom line and right line not counted. If the cells are touching this or this or are completely present in this small square, then only these are counted. And we start from left to right, and then we go downwards and again start from right to left. In this manner, we go and keeps on counting the cells. And we count cells present in all all of these four chambers, all of these four squares, and we don't count this cells touching this. If the cell is like present this, so this won't be counted in this square, but it will be counted in this one, because we count cells which are in the left and the above lines. Cells touching the left line and the above line of the square are only counted. And how to calculate the dilution factor. So when we take only 0.5 ml of blood and the rest 10.5, because it's 11 na, so 0.5 ml is blood and 10.5 ml is the diluting fluid. But we then discard 0.5 ml, we discard initial 0.5 ml of the diluting fluid. So the dilution factor is 0.5 ml of blood and 10 parts of the diluting fluid. So this gives us 1 part of blood and 20 parts of the fluid, giving us the dilution factor of 20. So calculation of the volume of WBC square. Since this is 3 mm and this is 3 mm, so and these are equal squares, equal length squares. So this is 1 mm, 1 mm, and 1 mm, and this is also 1 mm. So the volume will, and the depth which is shown here is 0.1 mm when we place the cover slip. So the volume will come out to be 1 into 1 into 0.1, which will be 0.1 millimeter cube is the volume of single square, and the volume of all four squares will be 4 into 0.1 millimeter cube, which will be 0.4 millimeter cube. Okay, so like volume of area in which WBC is counted in four squares, 1 into 1 into 0.1 into 0.4 is equal to 0.4 mm cube. And let us assume that the number of RBCs in all these four corner squares is n. So the number of WBCs in 1 mm cube will be n by 4. Are you getting this? Uh, we have assumed that the number of RBCs that we counted in all the four corner squares is as n, and the volume was 0.4 mm cube. In 0.4 mm cube, we counted n RBCs, n WBCs, sorry. So in 1 mm cube, by using the unitary method, we get n by 0.4 as the number of WBCs. And by multiplying it with dilution factor, we get the number of WBCs per mm cube equals to n by 0.4 into 20. We get n into 50. So, or where n is the total number of WBCs counted in the four corner squares. Have you got this calculation? Everybody?
Yes, sir.
First, we calculate volume of one square. This square, its volume will be 1 into 1 into 0.1. 0.1 being the depth, this depth. Volume is length into breadth into height. Now, this is basically height, and this is length, and this is breadth. So, volume of 1 will be 0.1 millimeter cube, and volume of all four will be 4 into multiplied by 0.1, it's 0.4. And in 0.4, we counted n cells, n WBCs in 0.4. And in 1 millimeter cube, it will be n by 0.4. Multiplying by dilution factor, we get n by 0.4 into 20, and n into 50 is the number of WBCs in 1 millimeter cube. Now, there can be various sources of error. It can be due to collection of blood, errors in collection of blood, due to pipetting error, or due to filling of the chamber. Blood collection errors may be due to prolonged tight application of tourniquet, might lead to venous stasis and leading to false elevation in the cell count. Excessive squeezing of finger puncture may lead to dilution of blood with tissue fluid, and inadequate mixing of blood with anticoagulant may lead to clot formation, which might lead to falsely low count. And if the blood is not mixed in the EDTA oil with the anticoagulant, there might be falsely low count. Errors in pipeting may be if we use a wet, chipped, or dirty pipette, we might get a false result, or use of a improperly calibrated pipettes, or not wiping the blood adherent to the outside of the pipette might give us false results. Now, due to filling of a chamber, errors can be incorrect filling of the chamber or spillage into the moats, which are the gutters, side gutters, or not allowing 2 to 3 minutes for cells to settle after the charging of the chamber. And if the our preparation has dried, then also it might give us false results. And if not using a specified cover glass, the cover glass which we use is a specific cover glass, not a normal cover glass, which is used in charging the Nubus chamber. And if we have to use a clean Nubus chamber, if it contains dirt or bubble, then also we might get a false reading. And various other uses of Nubus chamber are, it can be used as the RBC count, platelet count, CSF count, and sperm count. For RBC and platelet count, we use this central square is used in RBC count and platelet count. Anyone have any doubts? Hello? Hello? Hello?
Yes, sir, you are audible.
Okay. Everybody understood? Do you want me to repeat anything?